I also consider it a big privilege to talk to you today, particularly in my new hometown, which is Stockholm, for a year now. When we started the study, I was still in Germany at the University of Heidelberg and the German Cancer Research Center. Now, I'm very happy that we talked about colorectal cancer before we now are turning to pancreatic cancer for one reason, which I think is very, how should I say, illustrating the problem. There are 10 times more patients with colorectal cancer, but if it comes to cancer-related death, the number 9, which is pancreatic cancer, then really becomes the number 4 of cancer-related death. So the pressure on us physicians to really do something about this cancer is really high. And if you step back and look what has been accomplished in the last 10 to 20 years, and you again compare colorectal or even breast cancer to pancreatic cancer, then these days with colorectal and with breast cancer you can almost survive, but you cannot survive pancreatic cancer the same way you can survive colorectal or breast cancer. I mean, people with colorectal and breast cancer live 10 and more years, and then literally everybody who had been diagnosed with pancreatic cancer is dead by then. So, what we have put into the clinic in the phase 2 study, and that's what I would like to talk about here, and you will get the handouts of the presentation after this session then, is a new drug delivery system which is also targeting the vessels. I show you a picture on that respect in just a minute. So in other words, it's a formula of lipids, and you can put things into these kind of lipids and then bring this directly to the tumor. This is just a schematic diagram of a blood vessel, and here's the tumor, and you may know that once tumors are growing beyond 1 to the 9 cells, which is basically 1 milligram of tumor cell mass, the diffusion of nutrients and also of oxygen is not sufficient. So at that time, the tumors start expressing factors such as VEGF here to really attract the new angiogenesis, that is the new formation of vessels sprouting of existing vessels in the direction towards the tumor. Now, you are certainly familiar with, for instance, antibodies against VEGF or the receptors like Erbitux, which are targeting single molecules in this area, which, as you know, has proven to be successful at least in some tumor entities. These liposomic particles now, they kind of go to the endothelium itself right here, being charged, and then stick to that. They are going into the endothelial cells and release the paclitaxel, which then together with the second drug, the gemcitabine, is attacking the tumor. So I have a two-component kind of therapeutic system here. One, which is taking away the blood supply to the tumors, and eventually another drug, which is a clear cytotoxic drug, as is the paclitaxel itself. However, paclitaxel has so far not been known to be very efficient in pancreatic cancer. So there have been some phase 1 data when we started out, and we then wanted to design a phase 2 study, and you see the layout here. It's a randomized prospective clinical trial with a rather large number of patients in 4 groups. So gemcitabine in pancreatic cancer is the quasi-standard. I think you're familiar with that. So that's where we take as a baseline and a standard. And then 3 groups with the Endotac, which has then, these are the equivalents of the paclitaxel 11, 22, and 44 milligrams. And the patients were treated twice a week for 7 weeks. And after these 7 weeks, we went on basically with gemcitabine. After roughly 100 patients, we put in an amendment which would allow the patients, after we have seen about the safety of the drug, which is rather good, that would allow the patients to carry on with the Endotac treatment till progress. So that's why we have kind of 2 data sets here. This is just the overall 200 patients. The black line indicates the gemcitabine control, which has a median survival of 7.2 months, which is rather good. So that's in the ballpark, but on the upper side of what is seen in other studies. And then it's a very nice correlation, dose-related, between the low dose and the high dose of the Endotac. Now, the median survival is not that much different, although it's better than the gemcitabine, but I would like to point out that the 12-month survival, the 1-year survival, really is between 20 and more than 30% of the patients. And that indeed is a very good number, which is reached by some other of the biological and targets, and that is, I think, rather promising, really. One should also say that the toxicity of the drug is rather good. We didn't have lots of problems there. I'll come to that a little bit later. And if you then look at all the patients and you look at the 6-month and the 1-year survival rate, then it's quite clear that there is a dose-related response here, and the response rates all over are 68, 72, and 81% in the treatment groups, and 63% in the patients who just received gemcitabine. Now, if you look at the patients who received more than one course of Endotac plus gemcitabine after this amendment, it looks a little bit different. The gemcitabine still is about in the same range. 7.2 versus 6.8 is basically the same. That's not statistically significant. And then the treatment group really go a little bit further to the right, so to speak. Now, the median survival, the 50% survival, is basically hitting the 1-year survival for the 3 treatment groups taken together. So for the low dose, it's 9.2. For the medium dose, it's 13. And then for the high dose, it's 10 months. So that's really a significant advantage and really progress in terms of treatment results with this kind of treatment. Taken together, in this phase 2 study, with a rather large number of patients, 200, that's a third to a fourth of what you would do in a phase 3 study, we could already show that there is an extended survival of the patients, particularly if you look at the 1-year survival compared to the standard therapy. It is a new mode of action, targeting to the vessels as such, and not a single factor, particularly the endothelial cells, which I think has at least shown some proof of concept during this phase 2 study. The study was already a multi-center and multinational study, which was carried out in many European countries, including Stockholm and Germany, of course. And yes, I would like to acknowledge that now. I think that was it already. Thank you for your attention.